Good morning and welcome to today's MPG primer session. I am very excited to introduce our two presenters today. Elizabeth Kiernan is a science writer in Broad's data sciences platform, and she's joined today by Anton Kovalsky, who is also a science writer at Broad's data sciences platform. He'll be our main presenter today. He did his PhD work at Case Western Reserve University, following undergraduate studies in physics at Brandeis and graduate work in material sciences at the University of Illinois, where he focused on fabricating solar cells and characterizing their behavior. We are so excited to have them both here today and to be able to help us learn more about how to best utilize Terra in our research. And so Elizabeth or Liz will be monitoring the chat. And so they'll be both posting questions and links there, as well as responding to any comments in the chat. And Anton will be walking us through. So thank you so much to both Liz and Anton for being here today.
Thank you so much. So we can go ahead and get started. Welcome everyone to the second day of the MPG primer on using Terra. So first, a couple of quick reminders. If you were here last week, then you may have already done all of these things, but I'll just remind everyone anyway. All of today's slides and materials can be found at our Broad.io link for this particular session. Also, if you haven't already done the 2 steps that you need to do in order to be able to follow along with the hands-on portion of today's demo, then you should go ahead and sign up for Terra. And that's very easy to do. You just go to app.terra.bio, and in the place where you click the button to log in, if you have never logged in before, there will be a few extra clicks to register your Gmail account, and then you'll be set to go. It takes about 15 seconds. And then if you want to follow along with today's hands-on session, you'll also need to be added to a billing project. And we have a billing project that we've set up just for today's attendees. And in order to get added to that, you should go to our billing sign-up form. And I believe that we're sharing that link in the chat. And if you just add your email to that sign-up form, make sure that you add the email that you used to register for Terra, because those emails have to match. And if you do, then you'll be able to play around on our billing project for the next 2 weeks. In addition to that, I'll just remind everyone the logistics for how we're running this primer. We're going to use the chat feature in Zoom to ask questions. So just go ahead and post your question in the chat, and the moderators will either answer it in the chat, or if you want me to slow down and go back and show something again, then just post about it in the chat, and one of the moderators will chime in and ask me to reiterate something. You can also contact us with any questions asynchronously by emailing dsp-workshops@broadinstitute.org. So as a quick reminder of what we went over last time, we had a Terra overview. So we did a little overview of the why Terra exists and what it does. And we gave a tour of the platform, just an overview of the various pieces of functionality. And we had a brief hands-on demonstration of just cloning a workspace. We're actually going to do exactly the same thing today in terms of cloning the workspace. That's what we're going to start with as soon as we get to the hands-on portion. And then we'll move on to today's goal, which is to use an interactive analysis application. In this case today, we're going to use a Jupyter notebook, although there are a few interactive analysis applications available on Terra. And then next 2 weeks from now, at the next session, we'll talk about how billing works on Terra's cloud infrastructure. And then we'll have some time for a wrap up and questions and to talk in more detail about resources. So what we're going to start in a few moments is cloning the workspace. And if you cloned this workspace at the last session, you can go ahead and use the same clone. You don't need to create a fresh one for this demonstration. And as soon as we do that, we'll also launch our notebook. And then I'm going to just talk through the platform a little bit while the notebook starts up, because that process takes between 3 to 5 minutes. So we don't want to waste that downtime. So we'll go to the clicking, and then I'll come back and explain why we did the clicking. So as a reminder, or for anyone who wasn't able to come last time, in order to clone a workspace, and I'll demonstrate all of this in a moment, we go to the workspace, and you can either grab this link from the chat or from the slides, or you can find it in the way that I will show you how to find it in a moment. You click the vertical 3-dot icon that's in the top right, select clone, fill out the little modal that asks you for the details of your workspace, which basically means you need to give it a unique name so that you're not giving it the same name as someone else is giving their workspace clone. You need to select the billing project, which if you already have a billing project that you're signed up for, use whichever one you want. And of course, we've provided you with the workshop MPG 2211 billing project for today's demo. And then once you select the clone button at the bottom of the dialog box, you'll see that it's working. And after that, we'll move on to running the analysis in the Analyze Height GPA Jupyter notebook that is in the Analyses tab of that workspace. And so there are instructions, there's some instructions in these slides, and you can find all of the materials in the Broad IO link. So with that, I'm going to move over to the hands-on portion of this demo. Just a moment. There we go. And I'm going to give everyone a moment to get to where I am right now. So go ahead and go to this URL, app.terra.bio. And once you're there, you can log in by clicking on this main menu button. So over here where my name is, if you aren't already logged in, if you don't already see your name, then there'll be a button to log in. That's also where the button to register is if you haven't already done so. So I won't go through all of these menu items because that was what the last session's platform tour was about. I'm going to focus on just a couple of things that can be found in this menu. The first place I'm going to go is to my workspaces section. Before I do that, I just want to point out that later on, I'm going to also come to this cloud environments section in order to see the cloud environment and the persistent disk that I've spun up. And I'll also be able to pause it or delete it from this location. And it's just a convenient place to be able to see a list of all of the cloud environments and persistent disks that you've created. So, for now, I'm going to go to workspaces so that I can find the workspace that I want. There's a nice convenient panel up here for recently viewed ones, but I want everyone to see how to actually find a workspace that you know the title of. So I'm going to type in T101 because we want to use our Terra 101 Quick Start workspace. And you'll notice that there's a few different tabs along here. My workspaces refers to all of the workspaces that I have owner access to. There's also several levels of public workspaces. There are highlighted new and interesting ones, none of which are named T101. There are featured workspaces, none of which are named T101, but there are a couple of public workspaces. that are available to everyone with the T101 label in them. So we're going to go to this first one. And we're going to go ahead and before I talk at length about the workspace, we're going to clone it and start our cloud environment. So before I do that, I'll just point out that right now I can't do anything in this workspace because this workspace is locked. We're looking at the public read-only version of the workspace, so you won't be able to do anything in this workspace. You can't start a cloud environment. You can't edit the dashboard. You can't do any of the stuff that costs money. In order to be able to do that, you have to clone your own copy. So I'm going to do that right now. And I'm going to do it kind of slowly so that everyone has time to follow along with me as I'm doing it. I'll call this MPG demo AK. And again, this has to be a unique name per billing project. So there can't be multiple workspaces with the exact same name on the same billing project. So I'm going to stick with the my billing project, but you should all click here and select your demo billing project. And then you don't have to change anything else. There's a selector for the location of the bucket, and most likely no one here will ever have to deal with that. You don't have to change the description. You don't have to select an authorization domain. The authorization domain, if anyone's curious, is basically like a security badge. It functions based on the groups section of Terra. So you can lock a workspace behind a group, and then only people who are in that group will be able to clone the workspace. But we don't have that security feature enabled on this particular workspace, so I'm just going to click clone workspace, and you'll see a little spinny icon, and then momentarily you will be in your own version. So now our workspace locked indicator over here is gone. You can see up here the name of the workspace is now the name that I just gave it. And over here, you'll notice on your cloned workspace that instead of saying help-Terra, it will say the name of the billing project that you selected. So I selected help-Terra for this clone that I made, so that's why it says that here, but you'll see whatever billing project you selected. And now we're just going to go ahead and start our cloud environment. And we're just going to go ahead and do this clicking. And then, like I said, I'll go back and give a little bit more context. So what we want to do is just start a Jupyter environment on the default settings. So we click on this cloud icon that's part of this right-hand navigation bar, click on settings under the Jupyter section, and then just click create under use cloud environment. And we'll talk a little bit about what the default settings are. They were listed right there. And we'll take a look at what settings are actually available and how those compare to the default settings in a moment. So now that we've found our workspace and we've cloned it and started up our interactive analysis virtual machine, we can take a brief look around this workspace so that we see what it is that we're going to be analyzing today. So our goal today is to analyze the correlation between height and GPA of two sets of students. So this is obviously not a bioinformatic example. It's just a little toy example to demonstrate how Terra is wired together. And so you'll see that we have some data, some dummy data in the data tab. We have two data tables. We have the small student cohort, which is comprised of 8 students. And then we have a larger cohort comprised of 86 students. And we're just going to show you a notebook that takes in all of these data, basically takes in any GPAs that you want, and their heights, and generates some pretty graphs to demonstrate what correlation there is. And the idea behind the toy example is that we, for some reason, coincidentally see a correlation between height and GPA when we look at an undersampled cohort, but not when we expand to a properly sampled cohort. If we have time later on, I'm also going to demonstrate how to use the terminal in order to do things like look around inside of your persistent disk, or copy things in or out of it. So I just want to take a quick moment to point out that this button over here, the Files tab down here, will expose the workspace bucket. So I think this was probably covered in the first section, but in Terra, there are two storage locations. One is the workspace bucket, which is created when you clone a workspace, and the other is the persistent disk that is created when you create a cloud environment. So I've created a cloud environment. You'll notice it's still in the creating mode. When I mouse over this, it says that it's in the middle of creating, and that takes about 3 to 5 minutes. And once it's created, I would be able to open the terminal view and copy files into that location. And if I have time, I'll demonstrate doing that, but I'll just show you that there's a really easy way to copy things from your workspace bucket to your persistent disk by just clicking on. So when you are looking at your workspace bucket in this view, if you click on a file, it will actually give you a one-click copy, full command for copying something via GSUtil. It even includes the helpful period at the end so that it gets copied to whatever is your current location. So hopefully we'll have time to play around in the terminal at the end of this. What we're going to focus on today is using this Analyze Height GPA notebook. Our cloud environment is running, so I'll start this up in a moment. And then just as a reminder, these two sections aren't applicable to today's demo, but this is where you run batch workflows, and the job history tab is where you would see a history of your workflows. But today we're going to just launch some notebooks against this data. And as a last comment, I just want to say that these data are on the back end, they exist as a TSV, and you can download that TSV using this button, right? Download it as a TSV. I'm not going to bother doing that, but that's a useful thing to understand as we're going through this notebook. And you'll see why. That's because we read in the TSV as a data frame in the notebook. So I clicked on the notebook, and right now I'm looking at the read-only preview version. So this read-only preview is available to you regardless of if you even have anything other than viewer access to a workspace. So you can go to a public workspace without cloning it. Excuse me. And you can go to the Analyses tab and click on any notebooks that are in there, and you'll be able to see the read-only preview, which is nice from a public workspace perspective, but it's also useful when you're in your own workspace, because you don't always want to open the wrong notebook, and then you have to kind of wait between in the downtime if you realize you opened the wrong type of analysis, and now you have to update your cloud environment because you opened the wrong notebook, and so you chose the wrong settings. So you can always see this read-only preview and kind of scroll through it. It has whatever were the last outputs saved in the read-only version. But in order to be able to actually run compute in this notebook, you have to click either open or playground mode. Before I do that, I'm just going to run through what I said I was going to demonstrate with regards to the different settings for your cloud environment. So for this particular notebook, we only need the default settings. That's why at the beginning, we just went ahead and clicked create on that nice, convenient default settings button. But if you were to choose customize, which was a button at the bottom of the modal, then you would be taken to this view. And here you can select all of the settings for your cloud environment, and also get a kind of real-time cost estimate of how much it will cost for this cloud environment to be running, how much it will cost while it's paused. So while the cloud environment exists, even if it's paused, if you don't delete it, then just for the privilege of storing those settings, you will be paying a certain amount of money for that kind of passive storage. And also there's a certain amount of money that you pay for the persistent disk storage. So again, this is the separate storage location that's just for your interactive analyses. And you have basically 3 categories of things to choose from when you're setting up your cloud environment. You have your software, you have your computational power, and you have your storage. So software, which we refer to as your application configuration, is based on a set of Dockers that we've made available that are, they come pre-installed with a bunch of commonly used sets of packages. So you can use the default environment, which is the one that we're using right now. Or if you're interested in using Bioconductor or Hail, you can select those. We also have settings for, we also make available Dockers for some of our legacy environments. And that's just in case you have an analysis that for some reason it's not working all of a sudden, even though it used to work. So maybe the problem is that your analysis still relies on older packages. So you can actually select a legacy environment to check if that's so. We have some community maintained Dockers. For this one is for Pegasus. And if you're interested in having a community maintained environment that you want to share with others publicly, then you can always contact our team. And of course, you can also, if you're fond of working with Dockers and you want to Dockerize your own environments, you can click on custom environment, and then you just have to have your Docker on either Docker Hub or the Google Container Registry, and then you point at that location like this. So that's how the software selection works. And then you can select a bunch of different things for your computational power. This is called the cloud compute profile. And so you can select different numbers of CPUs and memory for those CPUs. And you can see up here our price estimate, our cost estimate for running this notebook is going up. You can also enable GPUs if you're interested in doing something like, why isn't this button working? Seems to be bugged out.
Anton, I think it's because you have your runtime environment running right now. Is that correct?
Oh, I can't. So, but this should be.
Is it created already? Sorry.
It is created.
Oh.
I thought you could click update, can't you? Or I have to pause it. Oh, do I have to pause it? Oh, it's update. So I think you can't. Oh, yes, that's right. So for all of these other settings, I could change it on the fly and then click update. Right. So before, oh, yeah. Oh, before this was changed, I could update this to have a new configuration. But for GPUs, you have to fully pause your environment or possibly even fully delete it in order to recreate. I think you can't add on GPUs to an existing cloud environment that didn't have them before. So yeah, I could click on this if I was starting this cloud environment from fresh. I can do something like this, and you'll notice when I do that, that's when my paused cloud compute price estimate changed. And finally, there is the persistent disk. And that's the last thing that you can kind of select in case you expect that you'll need more space. You can always increase your persistent disks. So that's why my cost estimate for my persistent disk storage went up, because I increased this by a factor of 10. Oh, and you can also change your auto pause settings. So auto pause is very useful. You probably should not turn it off, but you can always increase the amount of time in case you are using your notebook for something that's very time consuming and you don't want your notebook to time out in the middle of it. You can, if you really want, you can disable auto pause, but you should be careful not to forget that you have a running cloud environment, especially if you've pumped up the settings on it. So that is how we customize our cloud environment. But in today's demo, we're not going to bother customizing our cloud environment. This 7 cents an hour default cloud environment is going to do just fine. And we're going to go ahead and open this in an editable mode. I just want to point out that there's only a slight difference between editable mode and playground mode. Playground mode basically just doesn't auto save anything that you're doing. Whatever happens in playground mode stays in playground mode. And the main reason that we implemented playground mode was to cater to people who want to collaborate inside of the same workspace. So if for some reason you want to give someone can compute privileges in your workspace, but that they can't clone the workspace for themselves, so you want multiple people working on a notebook in the same workspace on the same billing project, then only one person will be able to open it in editable mode, that in the mode that auto saves everything. And the reason for that is so that people aren't overwriting each other. And it's basically a first come, first serve type of a setting. So I'm going to click it. I'm going to click open, and that opens it in edit mode. And if I were to add someone using my share button to this workspace, and I gave them can compute, but they can't, they can't clone the workspace, then if they tried to open this notebook while I was working on it, they would only have playground mode available to them. So they could still play around in it, but they would need to save their work somewhere else. So I'm just going to give a short rundown of some of the functionality of our Jupyter Notebook implementation, and then we'll just go through this notebook. And I'll pause at certain points to give people a chance to play around with the different aspects of it. So first of all, I want to point out the difference between the two types of cells. If you're familiar with Jupyter Notebooks, then you'll be familiar with the fact that there are markdown cells and there are code cells. So be careful if you inadvertently switch a markdown cell to a code cell or vice versa, because then all of a sudden it won't work. And that's, you can change a cell to be a markdown cell or a code cell using this little drop down, right? So I could change this into a code cell, and now it'll try to run this markdown formatted cell as if it were a Python cell, and it will fail because it is not a Python cell. So I'm just going to change it back to markdown, and now you can see the unparsed markdown. So I'm just going to go ahead and run this cell, and it will regenerate the outputted markdown. I'm going to do that with this button. There you go. So that was me running this cell. There are a few ways to run cells, and most of the time it won't be that you'll want to use this button, because the majority of people like to use the hotkey solution, which is shift return. So I can just shift return through all of these cells as much as I want. In addition to that, there are some fairly convenient run cell options under this cell drop-down menu. So you can run a set of cells you've selected. You can run all the cells below or above the cell that you've selected, or you can just run the entire notebook all at once. In addition to that, you can clear outputs. So you can either clear the output of the current cell that's selected, or you can clear all of the output in the whole notebook. And I'm going to ask everyone to do that because we have outputs already created here, and we actually want to watch them get generated. So we're going to clear all of our outputs. Just before we do, I want to point out that one of the other things that will be cleared is the number that was inside of this bracket. So just before I cleared that, there was a little number 1 in this bracket, and on the following code cell there was a number 2. This number refers to the number of times a code cell has been run since the last time that an output was either cleared or that the kernel was restarted. So with that, I'm going to point out where we do things like restart our kernels and where we can change the kernel. So the kernel that we're using for this notebook is a Python kernel that's indicated right over here. And if I wanted to change that, I could change it to an R kernel or whatever options you see here. We have additional kernels. So if you want to run an R notebook, you can do that. All you have to do is change your kernel in the middle of your analysis. So if you really want, you can have a notebook that's full of both Python and R, and some of those cells will work with one kernel and some will work with the other kernel. And the last thing that I'm going to point out about the kernel is you'll notice when I run a cell, so I'm going to go ahead and run this cell. That's the first cell that we have to run. And this is just your standard run-of-the-mill loading our necessary packages for this analysis. I'm going to hit shift return, and you'll notice, first of all, that this bracket will have an asterisk inside it, and this circle will have will turn into a dot. So when I mouse over this right now, I get a little indicator that says kernel idle. And if I manage to mouse over it before this cell stops running, while this dot is full, it'll say kernel busy. I'm going to go ahead and hit shift return. Kernel busy, kernel idle. So there we go. We ran our cells, or we ran our cell, and we loaded these packages. So we can go ahead and run through the rest of the notebook now, and I'll kind of give some medium level detail about all of the things that we're doing. Feel free to read over all of the detailed markdown that we've set. It gives lots of helpful context for why you do certain things certain ways in notebooks. This is a fairly typical way to do things in our in Terra notebooks. This is where we set our environment variables. And all this does is create a variable called project, and one called workspace, and one called bucket, and set those to your current workspace and your current billing project in your current bucket. So from now on, after running this cell, all I'll need to do is type in the word project, workspace, or bucket, rather than having to, for example, anywhere where I need to send something to my workspace storage bucket, I don't need to go grab this address anymore. I can just type in workspace bucket, or I'm sorry, I can type in bucket, and then it'll just work. And the same thing for project, and the same thing for workspace. So once we do that, we're going to go ahead and load in that data that we were looking at in the data tab into a data frame. So I'm just going to point out what some of these parameters do. So we're going to use a package called FIS, which is our FireCloud Selector Service package. This is a package that was developed in DSP a long time ago for exactly this purpose. It's a way for Terra to interact with other things via API, and to interact with itself in this particular case. And we're going to use a FIS function called getEntitiesTSV. So we're going to grab the TSV that, excuse me, my cat is attacking me. We're going to grab the TSV for the small student cohort. And so here you can see we use the project variable that we created in the previous cell, and the workspace variable we created in the previous cell. And then we're going to take the SM-student data table. That would be this one over here. And we're going to load it into a data frame. And I'll also just point out that it's useful to have this model parameter set to flexible. The reason for that is because if you don't, then the command will always expect that the genomic data model that you're using is named either participant or sample or pairs table, because those were the standard genomic data models that we originally created. But in this case, we're looking at something called the student model. And so we want to use the flexible mode. And then we also are going to rename one of the columns just for our convenience. So I didn't open the TSV of our data set. I pointed out how to download it as a TSV. If you were to do that and open that in a spreadsheet editor, you would see that the first cell of the first column has a very specific formatting. This is the formatting, and just FYI, if you want to create your data sets using a spreadsheet editor from scratch, you absolutely can. You just have to make sure that your top left cell is formatted in exactly this way. It has to have the word entity, and then colon, and then whatever name you want, and then underscore ID. But we're going to change that for this data frame. So here we've created this data frame, and then we're going to do a data frame rename one of the columns, and we're going to tell it to rename the column that has this complicated name to have the simpler version of the name. I'm going to run this, and then I'm going to take a look at it. And there it is. That is our data from the data tab. So it's a beautiful way to be able to just look at whatever data that you have on the fly as you're doing your interactive analysis. So once we do that, we're going to go ahead and do some analyses on all of these data. And we're just going to run some plotting functions. So the first thing we have to do is run this cell that defines all of the plotting functions that we want. Again, this is fairly standard notebook work or fairly standard Python work, however you want to look at it. And then we're going to start by looking at some of these samples. Right, so when I run this cell, I get a nice plot of the height distribution for this particular subset, right? And that's the subset that I've defined up here, right? I decided to name this particular small student subset as subset. And so I can look at their height distributions. And then here, so in a moment, I'll pause and I'll let everyone try out some of their own, whatever you want. You can look at one of the other variables. You can try to figure out how to read in the larger cohort. Although, spoiler alert, this is how we read it, the larger cohort, right? So instead of naming it subset, we've named it data set. So I'm just going to go ahead and show you how to how some of these other plots look, and then we'll pause for, say, 5 minutes and let everyone just play around. So I've done the exact same thing here. I've loaded in a data set, or I've loaded in the larger cohort, right? The one that just is called student rather than small student. I've named that data frame data set, and I'm doing and I'll do a quick little rename so that that first column isn't something funky, and then I'll take a look at it. There it is. And then we can go ahead and do things like plot their height distributions. We can plot their height versus cumulative GPA correlations. And you can see here, when we look at the larger student cohort, then it doesn't appear that there's a correlation, which is as you would expect it. Whereas when we look at the small student population, there is some sort of a correlation between height and GPA. And that's, so the toy example here is, do we, does our data give us pause? Because obviously this is a correlation that you wouldn't expect.
Hey Liz, yes, I can hear you well. I do think that we froze and then lost Anton. Looks like he'll be coming back soon.
Great. Well, this is actually a perfect opportunity then to ask everyone if they need any part shown again. We definitely want to make sure that everyone in the audience today is able to access the workspace, clone it onto our free billing project, and start their VM up. Again, you have access to this billing project for a week. Longer if you email us and request it, if you want to try to save some of the work that you've done. And that billing project enables you to try anything else that's public in Terra as well. So we definitely want to make sure everybody feels comfortable, getting up and knowing how to make a copy of a workspace that you can work in. Does anyone have any questions about that? Please feel free to post in the chat, or need me to show anything that, show anything that Anton was demonstrating.
Liz, I was wondering if there's certain public workspaces that are kind of more vetted or kind of good places to start. Like if you were going to start with RNA-seq or something straightforward, do you have any advice for choosing a workspace?
I do. I was not prepared to share my screen, so I'm closing up 1,000 windows right now, and then I'm going to share my screen and just show people where to go to find that sort of information. And I think I'm ready. All right, let me see really quick. Okay, terminate. All right, so let me just share screen, and I apologize if anything kind of funny comes up here. All right, there we go. Okay.
No problem at all. And we're nearing the end of this portion anyways, and everyone got such a great start. I think we'll be able to follow through and understand why height and GPA are not a direct correlate. It's so interesting to see though here.
All right, can everyone see it? Am I sharing my full screen? Can everyone see this okay?
Indeed. Yeah.
Okay, perfect. Great. So I just went back to the main page here at app.terra.bio. And I am going to show you something that Yasha showed in the first session, but there is this library dropdown in here. And within this library dropdown, we have something called featured workspaces. So these are curated workspaces that the Terra team curates themselves, which means that they're going to have downsampled data in them so that you can try the different tools within them at fairly low cost. There's also usually cost estimates in there. So if you want to try it on your own data, you can get a sense of what it'll be per certain gigabyte of data. And you can search across these workspaces. So you mentioned, what if you're interested in single cell? Is that right? Or RNA-seq?
Yeah.
So you can type in by keyword here, single cell, or even single cell, and come up with a list of workspaces that include single cell analyses. Or you could type RNA, for example, and see if there's any workspaces that come up for RNA-seq analysis. It helps to put the space in there. You can also search by filtering on the left over here. So you can see we have certain facets that you can select. We have getting started, but we also have analysis tools or experimental strategies. So if you're interested in RNA analysis, you can click on that. And then we have, 8 workspaces that are curated towards RNA analysis. You can also further filter by I believe tools. Did I pass that? Yeah. So if you were interested in Whittle workflows or notebooks for a particular analysis, you could search by that too. So I think that's pretty much what I want to show you there. And just in time for Anton to come back. So hi, Anton.
Hi. So I'm back on my phone because my internet is still wonky. So you may have to continue. I wanted to apologize. I hope that I was not cut off until I finished showing the notebook.
Yeah, we got most of the way through, which was perfect. And I think everyone was, on the notebook and making it through. It looks like the part just after you spoke was that if that lesson of if you look at 8 samples, you see the strong correlation. And
right.
Once you get to the full data set, you see that you were led astray by those 8.
Great, wonderful. As an aside, I'm really annoyed at the Google Home. It's Google Fiber. This is supposed to be the good internet.
No, well, thank you both so much for your time today. And we really look forward to your 3rd session that will happen after the Thanksgiving break.
Thank you so much, Sarah.
Okay, have a lovely day, everyone. Have a wonderful holiday.